go across the cell membrane, even with channel and even with pumps. Okay, so for instance, hormones. Okay, so large molecules. Okay, large molecules. Okay, all right. This one we got transport of what? Transport of very large molecules. Okay, and because they are large, they can actually go through the cell membrane. Okay, and so in this case, like that, they form some sort of vesicle, and then these vesicles would then transport the molecules by fusing with the cell membrane and then eventually release. Okay, all right, good. So for instance, if you take something like azocytosis, classic example of azocytosis is the word, that is the release of word. Let's say release of enzymes. Enzymes are quite big molecules. Release of neurotransmitters. Okay. All right. Release of neurotransmitters. Okay. So we have what? We have okay, so these ones, they are large molecules. Okay, so large molecules like that. So enzymes, so this is azocytosis. Okay, so azocytosis, okay, that one, molecules are coming out. So you can see from my diagram here, things are coming out. So after the formation of the of these molecules, okay, they will be in vesicle. The vesicle will come and fuse with the cell membrane, and eventually it will be released out. And then endocytosis is a reverse of it. Okay, so large molecules, it could be drugs, it could be certain hormones as well. Okay, some hormones are coming into the cell. They are large molecules, and so for large molecules, for them to come into the cell, okay, they are going to be where? They are going to be, for them to come into the cell, okay, they will bind to some kind of receptor, and eventually they will just activate them and then join, rush into the cell. Okay, all right, great. So in this case, basically they just bind into the cell membrane and then they get into the cell. So this side is the word is azocytosis, something coming out of the cell and something getting to the cell. And then for the azocytosis, I give this one. This is a classic example of azocytosis. This one, the release of neurotransmitters. You can see the neurotransmitters are in a vesicle. Okay, so they get to the word. This is the axon terminal, and eventually it will be released. Okay, to be released from the axon terminal, okay, into the synapse. So this release of neurotransmitter is a big molecule, so it can diffuse across. It can diffuse or even go by active transport across this membrane here. Okay, so what they are going to see, that some molecules can diffuse. Okay, if you look at this one, okay, this one like that, it can diffuse. This one you can see is going through channel. So this is facilitated diffusion. But this one, like this neurotransmitter coming here, it can't. It's very large. And so to form a vesicle, and then it will eventually burst, okay, like that, fuse with the cell membrane, burst and release into the synaptic cliff. Okay, so we have this. That's a neurotransmitter. Okay, that's a neurotransmitter here. Okay, like that. Okay, great. So it's a large molecule. Okay, so and that's basically what happens. Okay, so when it comes to endocytosis, molecules get into the cell. We can also get two types. Honestly, we can technically there are two types, but we've added one to it. Okay, and this side, okay, the one we've added is so here when the solid molecule, okay, is solid molecule like this one, and when it comes to solid molecule, that's the phagocytosis. That's the mechanism that amoeba tend to use to feed. Okay, so they surround the particle with the word with the cell membrane, and eventually we form something we call phagosome, which is a food vacuole in this particular case. And this one like that is what that's a phagocytosis when it is solid. And when it is liquid, it is what? It is a pinocytosis. And then sometimes the substance coming is what? It's actually receptor-mediated endocytosis. When you talk of receptor-mediated, it means that the substance coming is having some sort of you see here, look at that. So these molecules are coming, and then they will bind to a particular receptor. Okay, so when they bind to a particular receptor, then they will trigger the formation of what? The formation of the vesicle here. Okay, so this chemical substance will come, and then there are some special receptors here. So when they bind to the receptors, then eventually they will trigger the formation of the vesicle that will actually bulge in, okay, with a substance. Okay, so you can see these are the receptors here. Okay, these are some of the membrane proteins acting as receptors. Okay, so this one bulges inward, and then eventually will form the vesicle, and then the molecule will be transported inward. Okay, all right, good. So we have the pinocytosis being liquid, and then phagocytosis, and then we have a receptor-mediated endocytosis, which means that the molecule that actually came onto the surface of the cell membrane needs to bind to specific receptors. And then when it does that, it will trigger the formation of some sort of vesicle, which this molecule will enclose it and eventually get into the cell. Okay, great. So that is what we talked about this morning. That's what we ended with. Now, the last part we're going to talk about is the cell cycle. Okay, and when you come to the cell cycle, okay, cell cycle is particularly important. Okay, great. In a sense that I think we are quite familiar with this one that on my screen. This is cell cycle. Okay, when we talk of the cell cycle, the stages that the cells tend to undergo to complete their development, basically. Okay, so this my diagram here is a cell cycle that I'm talking about. Okay, and then this diagram. Okay, so when we take the cell cycle, we have we have the cell growth, we have the DNA replication, another cell growth here. Okay, great. So basically, this is what I'm going to walk you through, and then if time will permit, maybe I will do some basic structure of DNA, and then I can call it a day. Okay. All right. So when you talk of cell cycle, what is cell cycle? If I may ask, what is cell cycle? When you say cell cycle, what is it? When you say cell cycle, what is it? Cell cycle. Okay. When you say cell cycle, what is it? Okay. What is cell cycle? When you say cell cycle, what is it? Yes. All right. When you talk of cell cycle, what is it? Yes. Anybody from the audience? When you say cell cycle, what is it?
So okay, yes, I want to borrow it from it refers to the stages through which a cell passes and then replicates its DNA.
Okay, stages that the cell actually goes through to replicate its DNA. Okay. Well, I would have well, the replication of the DNA that you brought is what is making what you said wrong, actually incorrect, actually, because the replication is a complete this thing. It's a whole part of the cell cycle. Okay, all right. So what you said, so it is more or less a series of events. Okay, it is a series of words of events that the cell tends to undergo. Okay, so it is actually, so cell cycle is the word, is the series. Okay, is the series of events that the cell undergo. Okay, undergo. undergo, undergo. And this word, and this comprises or its word. It's consists of words. So the cell cycle consists of word, consists of cell growth. Okay, all right, consists of the cell growth, okay, DNA replication, DNA making a copy of itself, and then we have the word, we have the cell division, okay, and word. So maybe the last part is the cell division. So, okay, division. All right, good. So in fact, these events, okay, growth, DNA replication, and then eventually the cell division. So it's going to divide. Okay, good. So these are the what? These are the stages that the cell tends to go. So what is here? What I have here is more or less, it's indication of more or less time. Okay, more of time. Okay, all right, good. So in this cycle, more or less, it's time allocation. Okay, it's not for a particular cell. It's more of generic thing that I picked online. Okay, good. So from here, so you can see that the cell will divide. The cell division takes just a small part of the word of the entire life cycle of the cell. Okay, but the bulk majority of the cell will be taken by cell growth. Okay, so from this point here, from the green, from this side, from here, okay, from this this green, light green, all the way to this light blue, that's the word, that's the interface of the cell. So basically, when we talk of the word, the cell cycle, so cell cycle, okay, cycle actually is what it is, the interface, interface plus word, cell division, interface plus cell division. Okay, that's the interface plus cell division. Okay, so because the interface will have the cell growth, the DNA replication. Okay, so this G1 is actually growth phase, G2 is also growth phase, and then the S phase is the what? Is the DNA replication. DNA is going to make a copy of itself before the cell is going to divide. Okay, so if we look at this one, if we look at this, okay, if you look at this time allocation here, so when we talk of interface, what is the components of the interface? So the interface actually consists of the word G1, which is the cell growth, and then S phase, which is actually the way the DNA replicates, and then the G2. Okay, so when we talk of the interface, these are the three main phases of the word of the interface. Okay, these are the three main phases of the word interface. All right, so from the G2, then the cell is now going to enter into the word, into the division phase, the cell division. So you can see that the cell division doesn't even take so long, but the growth phase is where the vast majority of the cell actually tends to be. Okay, great. Now there's something missing here that, let me pick another page. We could call it what? GO. There are some cells that do not divide. So those cells, when they get here, they are permanently, so let me go and get you cell cycle again. Okay, let me get you cell cycle image again, whereby, so cell cycle with GO. Okay, GO. Okay, so let me show you image of it. All right, so let's see cell cycle with GO. Okay, cell cycle with GO. Okay, cell cycle with GO. Okay, let's see what I can get. Okay, okay, great. So let me copy this one and then come and give it to you so that you have idea what I'm talking about. Okay, great. So if you look at this my diagram on the screen, okay, so this is still the cell cycle. Okay, so this is a cell cycle diagram. Now, if you look at this my cell cycle diagram, so you can see that when the cell is not dividing, okay, so there are some cells that are not dividing. Okay, for instance, the muscle cells, okay, once they are mature, they don't divide. So the muscle cells will forever and ever be in what, zero. Okay, that is why when you get infection with a muscle, it's very difficult for them to heal. That's why when people get polio when they are kids, they are forever deformed. Okay, when they get polio when they are kids, they are forever deformed. So when we have this, when we have the cell cycle, okay, the cell cycle here, okay, this cell cycle here, okay. So when the cell is not dividing, okay, so you can see from this diagram, so we have the G1, okay, the G1 here, which is the growth phase. Okay, I will talk a little bit about each of them, okay. And then the S phase is basically where the DNA is going to replicate. All the DNA in the cell is going to replicate. It's going to make a copy of itself, okay. And then here the G2 is more or less the word. It's going to make sure that is everything fine, is everything okay. Do you have some repair? Okay, remember the DNA is going to replicate it. The DNA is going to make a copy of itself. Is there any error there? It will check it. Do you have enough enzymes? Do you have enough protein? Do you have enough ATP? Because from G2, the cell is going to divide. So it is here that it's going to check that do I have enough of everything? Okay, before the cell will go ahead and divide. Okay, great. So you can see from, but when the cell is not dividing, it is permanently at what? Some sort of, it is being arrested, okay? It's not at rest too, okay? It's just, it will be in a state, there are some cells that are not dividing. So those mature cells that are not dividing, like the nerve cells, those mature nerve cells, they don't divide. So those mature nerve cells, they will be in what? They will be in a GO. The mature muscle cells will also be in GO. They won't go through this phase. No. Okay, once they become mature, they don't divide. Okay, so they will be here. They will be permanently here. All right, great. Now, so let's take the parts one by one and talk about them. So if you take so you can see that when the cell is not dividing, okay, so when the cells, okay, so cells that do not divide, okay, that do not divide at the word at the a phase at a phase called what? Called geo. Okay, so that's when the cell is not dividing, okay, like mature nerve cell and what? And mature muscle cell. Okay, great. All right, great. So once they mature and then they are not dividing, they are permanently at what? At zero. Okay, so they are in a state of, they are in the metaphase state called what? Zero. They won't go through here, they won't pass here and come to us, and then they are not going to divide. So they'll be permanently be at this place. Okay, great. Now let's look at something interesting here. Now let me start with the interface. Okay, interface. Now the interface, you see, that is where, okay, a lot of activities tend to occur. You see, SS textbook, SS GAST. I always have problem with that book, biology GAST in particular. They say that it's the resting stage of the cell, but no, that's very, very wrong. I always, so please, when you go to SS and you are teaching, please don't be using that book that much. That book has so many mistakes in it, and then back in the days, the mistakes there, they are still there. They've not corrected it. And they're even adding more mistakes. So be careful with SS GAST, biology GAST. Okay, great. So in fact, when you come to the interface is the most, in fact, it is the most metabolically, okay, metabolically what? Active phase of the cell cycle, Okay, it is the most metabolically active phase. but some funny books will say that it is the resting stage. No, it is not. Okay, it is the most metabolically active phase of the cell cycle. Okay, some books will say resting stage. Please, it is never resting stage. Why? So here it is the most metabolically active phase. It is divided into three phases. What are they? G1, G1, and in G1, what is going to happen in G1? G1, that is where most of the organelles, okay, the organelles, okay, are going to do what? Are going to replicate, okay, are going to replicate, all right? So are going to replicate. When you say replicate, means they are going to make copies of themselves, okay? They are going to, so the ribosomes there will make more, they will do like more of ribosomes. Some will not actually replicate, but they will just make more copies. So most of the organelles here are going to make more copies of themselves, okay? So mitochondria will make a copy of itself. If chloroplast is there, it's also going to happen. If the ribosomes is there, they will increase the number of ribosomes. Okay, number of ribosomes that will increase. The remember, assemblage of ribosomes occurs in the nucleolus, and then apart from that, they will do what? More enzymes. Okay, more proteins will be formed. Okay, more proteins will be formed. And when I talk of protein, we are talking about what? The enzymes, you know. Remember enzymes. So here the DNA is going to replicate. Even the DNA replication, there are so many enzymes involved. Okay, here, this phase of the life cycle, there are so many enzymes involved, plenty enzymes, and all these enzymes need to be manufactured, okay, in this particular, at the growth phase 1. So some books will call it gap phase, other books will call it growth phase, okay, whichever one you pick is absolutely correct, okay. Some people will call it growth phase 1, growth phase 2, some will say gap phase 1, gap phase 2. Whichever one you settle on is correct. So here we have this where the G1, most of the organelles are going to make copies. So to replicate something is to make a copy, So that's a copies, okay, copies. That's the meaning of the word replicate. So replica of something means copies, okay. You are going to make more copies of, okay, so more enzymes, more protein here, okay, more proteins will be what formed. So enzymes, okay, like enzymes and stuff like that, okay, that are going to be used, okay, that's where they are going to form more energy, ATP synthesis, okay, more ATP will be produced, more ATP, okay, so more ATP, okay, more ATP will be produced, okay, great, all right, so more ATP will be produced. So you can see that if somebody tells you that interface is a resting state, the person doesn't know what he's talking about, because a lot of activities tend to occur in this phase of the cell cycle. Okay, so there's no way it is a resting stage at all. It is actually the most metabolically active phase of the cell cycle. Okay, so the interface G1 is the where organelles are going to replicate, more proteins will be formed, more energy production in the form of what? ATP. Okay, will all occur in this particular place. Okay, all right, good. And then you see that so when they grow, okay, so you can see that they say that cell content will duplicate, okay, except what? That's excluding what? Excluding the DNA, the chromosomes. Okay, great. So everything is going to double up. Everything in the cell is going to double up. Okay, all right, great. And then if the cell is not something that it has the intention, okay, that is going to divide, then this cell will forever stay in this place, which is what? The GO, okay, to forever stay in GO. If the cell is not going to what? Okay, so here, if the cell is not going to divide, if the cell is not going to divide, then it will stay at what? At GO. Okay, so it will be arrested at what? At GO, like that. Okay, all right, great. So, yes, please.
With all due respect, I joined 5 minutes ago, and you are making use of G1, G2, and then S.
Yes, I can't go back. In fact, I waited for solid 30 minutes. I'm sorry, I can't go back. Okay.
Oh.
Let's move on. After you can watch the video. I'm sorry about that. Thank you.
Okay.
So we have the word. So we have the G1 here. Okay, that is the most. So that is a part of the. So from the whole of this point here, from the whole of this arrow, all the way. So from G1 here, all this arrow all the way up to this end here. That's the interface of the cell cycle. Okay, the whole of that's the interface of the cell cycle. Okay, good. And then this side of the cell cycle is where the cell divides. So this side is the nuclear division, the mitosis and nuclear division, and then it will be accompanied by cytokinase, which is the cytoplasm division, and then the cell is going to divide. Okay, great. So so from G1 is what I've talked about, what is happening in G1 of the interface. In the G1, what is going to happen is that most of the organelles, they are going to replicate. They are going to make copies of themselves. Protein, okay, more proteins will be formed. So that's a protein synthesis will be carried out. Okay, so we have the enzymes. Okay, so when you talk of protein, remember enzymes are proteins. Okay, so enzymes are protein as well. And then we have the word. So we have the enzymes here will be formed because even the DNA, for DNA to replicate, okay, we need a lot of enzymes to do that. And then we have the word, the ATP. More ATP will be formed. More ATP will be produced. Okay, more ATP will be produced. Okay, all right, great. And then, as I said, if the cell is not going to divide, it will stay in a phase, we call it what? GO. When the cell is not going to divide, to stay in a phase called GO. All right, good. Then after G1, okay, after G1, let me duplicate this slide here. So after G1, the next phase is the word. The next phase of the interface is the word. Is the G2, is the S phase. Okay, so after G1, the next phase is the word S phase. Okay, S phase. And S phase, the main thing that occurs at the S phase is the word, is something we call DNA replication. DNA replication. So that's the main thing that occurs here, DNA replication. And when we say DNA replication, what does it mean? It is DNA making a copy of itself. Okay, the cell has intention of dividing. Okay, so the cell has intention of divide. Now look here, this cell here is going to divide. Look at what is here. You see, after the S phase, you see the cell has what? You can see that the DNA here, when we started, is 1-1. But when it comes here, you can see that it has become, the red has double up. The blue has double up. Okay, so in the case of let me show you something here. Let me draw something here to illustrate what I was talking about. Okay, so here, all right, so let's say this is the chromosome like this. This is the chromosome like that. Okay, great. And then this chromosome, okay, like that. So usually they are arranged in pairs. So let me say there's another pair like this. And this pair, I think I've told you people before, that's a homologous pair. All right, good. So these are these chromosomes here. Okay, so before the cell would divide, these chromosomes that you see, which are homologous pair. Okay, it is homologous pair because we are diploid organisms, and so we've had one from our mom and one from our dad. Okay, so this is that. So maternal one and paternal one, like that. Now, so before the cell divides, what is going to happen is that there is going to be what? So remember, the DNA actually condenses to form the chromosome. If you could remember from our first meeting, okay, the DNA actually condenses to form our chromosome. Okay, to form the structure. Okay, so our DNA, remember, the DNA looks like this. Okay, the DNA, okay, looks like that, like that, like that, like that. Okay, like that. Okay, like that, like that, like that, like that. Okay, great. And then to condense, okay, by coiling around some round structure, we call it what? Histone protein. If you could remember that they coil around what? Histone. histone. Okay, they coil around the what? The histone protein. So when they do like that, they condense to form this structure here. Okay, good. And when they condense to form this structure here, this particular structure we are seeing here, we call it, or this condensed form, we call it what we call this one chromatid. So this one you are seeing here, okay, each one, this one is chromatid, this one is chromatid. Okay, so this is so what you are seeing here is what? It's a chromosome. But this chromosome consists of what? This chromosome consists of it's a one word. So chromatid. So it's a condensed form. Okay, so this chromatid. Okay, so this chromosome is one chromatid, another chromosome here, another chromosome here, also one chromatid. Okay, but before the cell will divide, okay, what is going to happen is that at the S phase, the chromosome there is going to do what? Is going to the DNA, or the DNA which from the DNA is going to replicate. So more or less the chromosome is going to double up. That's what is going to happen. This DNA will make a copy of itself. And when it makes a copy of itself, you see that, look at
Hello,
that. So
Doc.
yes,
Please,
please.
your screen is not showing at my end.
Let me see. Okay, I mean,
Check your internet,
please, it's your net.
No, it's okay.
Go
Maybe
out
it's coming
and
from
read.
Restart, restart your
Go
device.
out and
Restart your
rejoin,
device.
please. Okay. All right. Great. Okay. So here, if you look at if you look at my structure on my screen, you can see that this is my DNA. Okay, so this is the DNA, and then the DNA will coil around what? Histone proteins. And when they coil around the histone protein, they coil to form the structure you see here, okay, which is the what? The chromatid. Okay, so the chromatid is this highly condensed. So it's a chromosome. So this is actually a chromosome, but this chromosome consists of what? One chromosome. Chromosome. Okay, it's a chromosome consisting of what? One chromatid. Okay, but after DNA replication, so let's see. On just this one. Oh no, I can. Okay, great. So after DNA replication, I think this one we've already talked about. Let me compress this one a little bit. Okay. Okay, so we've talked about this one already. So this one can go up. Okay, so still on the interface. Okay, so let me increase the size a little bit. Okay, great. Okay, great. So after DNA replication, what is going to happen is that, so let me take this one up here. Now, what you see here like that is going to double up. The DNA is going to double up. Now, let's see how it's going to double up. Okay, so the DNA is going to double up. So you see this one here is going to become this. Okay, this like that. And then this like that. So the DNA will make a copy of itself, but they won't separate. Okay, and then let me pick the other one, the blue one. Okay, so before the DNA divides, so you see this one to also make a copy of itself like that here, and then like that there, like that here, like that. Okay, so before the DNA divides, so now you can see that after the word, after the S phase, what you are going to have is that we'll have the word, we'll have chromosome consisting of what? We have the chromosome consisting of sister chromatids. Okay, so after the the chromosome there, so afterwards, so after after S phase, okay, after S phase, the chromosome, okay, chromosome consists of what? Consists sister word consists of how many? It will have how many chromatids? It will have what? How many chromatids will it have now?
4 chromatids.
No, it
46,
will have. You can see each chromosome now will have what? Will have two what? Two sister chromatids. Chromatids. Have you seen it? But before that, you can see that if you look at before the DNA replication, okay, if you look here, before the DNA replication, you can see that one chromosome consisting of just one chromatid, right? But you know, after the S phase, the chromosome will double, the DNA will double up. So you see each chromosome will have what? Two sister chromatids. Please, are we clear with that? You can see that this one here has double, has become this. And this single one chromatid here has become double. Okay, so it's what? So each chromosome, okay, so the chromosome will what? Will consist of what? Now consists of the chromosome consists of, so each, let me say each chromosome, okay, so let me make it each. So each chromosome consists of what? Each chromosome consists of sister what? Two sister chromatids. Here it consists, each chromosome consists of one chromatid. This one here, one chromatid, this one chromatid. But after the DNA replication, when the DNA makes a copy of itself, which it occurs in the word, the S phase. So the S phase of the life cycle, the most important thing that happens there is the word, is the DNA replication. So the DNA will make a copy of what? Of itself. And when the DNA makes a copy of itself, now we'll have our chromosome not consisting of only one chromatid. Each chromosome will have a sister chromatid. The cell is going to divide. So you see that each of them now consists of what? Sister chromatids. Okay. So if I go back to this my diagram, you can see that you see the cell is going to divide. So this is the cell here. You see it's one chromosome each. Okay, one chromatid. Now when it gets here, still one chromatid each. Chromosome consists of single chromatid. But when after the S phase here, look at what has happened here. Look at the picture here. You can see that the red is now consisting of what? Two sister chromatid. You can see the blue has also doubled up. The reason it has doubled up is because it is what? It is the DNA here has made a copy of itself. Okay, the cell is going to divide. So it is good that the DNA makes a copy of itself so that when it divides, the half of each can go into each cell. Okay, so that's what is going to happen. So the DNA will make a copy of itself, and that occurs in the word in the phase of the interphase. Okay, that's the second phase of the interphase. Okay, the second phase of the interphase called the what? The S phase, and then the G1. The G1. Let me call this one is the first phase. Okay, good. All right, so that's the S phase. So S phase is predominantly DNA making what? The DNA. Okay, so its DNA makes what? A copy of itself. Okay, so the DNA. Okay, the DNA makes a copy of itself, and that is what we call replication. Making a copy of something is known as replication. The DNA making a copy of itself is called DNA replication, and to occur in the S phase of the cell cycle. And then the second part, which is the what? Which is the last part, the third part. Okay, so let me duplicate this slide again. Okay, so let me duplicate here. Now, so the third part of the cell cycle, which is the word. No, this one, I want to duplicate this one. Okay, so the third part of the cell cycle, okay, so this one can go now. Okay, so the third part of the cell cycle is the what? Is the G1. Okay, so the third part of the cell cycle, that is the what? That is the G1 phase of the cell cycle. The G2, the 3, so part 3 is the what? Is the G2. Okay, the G2 phase of the cell cycle. And the G2 phase is more or less a checker phase. It's like I want to find out that is everything okay with this cell? It's like is everything okay? like that. Okay, so all that I want to find out is, okay, I want to find out whether everything is okay. All right, I want to find out whether if everything is okay. So here, in case we don't have enough ATP, more ATP will be made. Okay, so here is more or less a checker point. You understand that? So like you see, you can see that double check everything. So here, we are double checking that do you have enough? Is there enough ATP? Enough ATP, because the cell is going to divide, and mitosis will need what? Energy. You have enough what? Proteins, or into bracket maybe enzymes. Okay, so we have a lot of enzymes involved in this process. Okay, do you have is the DNA accurate? The DNA after the replication here is the DNA replicates. So are the DNA replicates correct? Okay, are the DNA replicates correct? So you ask all these questions. Do I have enough proteins? If they don't have enough protein, they will make sure they will stop it here and they will manufacture more proteins. Is the DNA replication, is there any fault with it? If there's fault with it, the DNA, they will stop the DNA and repair the error before it will proceed. You see, as at this point, once it divides, nothing can be done about it. So to check, is the DNA replicates, are they correct? Do I have enough proteins? Do you have enough ATP? You understand that you have enough, so that's enough energy there, enough protein. Are the organelles, the organelles that replicated, okay, replicates enough? Okay, so they will be asking all those questions, okay, and if they are not, so the second part is more or less the checker, okay, more or less, so let me say more of what? It is more of what? Quality control, okay, more of quality control stage, okay, all right, so to check that the G1, what happened in G1, the whereby the organelles replicated, okay, here in the G1, do I, after the replication, is everything fine? Do I have enough? The G1, remember, protein will be synthesized, more protein will be synthesized in G1. Okay, they will ask here when they get say, do I have enough protein for the cell to divide? Do I have enough organelles? Because the organelles are going to replicate here. So they will ask, do I have enough of it? Okay, do I have enough of the organelles? No, except the nucleus. Okay, except the nucleus, all the other organelles are going to replicate here. Okay, and then at the S phase, that's where the DNA is going to work. It's going to make a copy of itself. You see, when it makes a copy of itself, as we can see from here, when the DNA make a copy of itself, you see that the two, they don't stay. When they make a copy, when this one makes a copy of itself, it still stays as one. It is still one chromosome, but this one chromosome is consisting of what? Of two sister chromatids. So they don't separate. Okay, they stay together. So they only separate at the what? At the mitosis. When they perform the mitosis, that's when they will separate and one will go. Okay, you can see that when they split this one, red splits in here, and then one went here, and then one went there, and then blue splits here, one went here, and one went there. Okay, great. So it is at this point. It is at this point here. At this point, at the G2 point, they will find out, do I have enough ATP? Do I have enough proteins? Okay, specifically enzyme. Do I have enough organelles? Do I have is the DNA replication, the replicas of the DNA, are they correct? Okay, if they are not correct, they will make sure that they will repair the error, okay, before they will continue. So the G2 phase is more of what? Quality control phase of whatever that has happened here, okay, anything, whatever sort of event that actually occurred here, they will double check whether everything is fine before the cell will go ahead. The nucleus will divide, followed by the cytoplasm, and then we get the cell division. Okay, great. So we know that interphase is a most metabolically active phase of the cell cycle. That is where we have the organelles making copies of themselves. That's where more proteins are produced, including enzymes. that is where more ATP will be formed. Okay, all right, good. And then afterward, we go to the S phase. S phase, the only thing that happens in the S phase is the what? Is the cell making, the DNA making a copy of itself. So before that, you see that the cell is consistent of just one chromosome here, the homologous chromosomes. Now, by the time it gets to after S phase, you see that the chromosome will double up in its amount. Okay, the DNA will double up in its amount. Okay, so you see that each chromosome will now consist of what? 2 sister chromatids. Okay, but before that, it's only one chromosome consists of 1 chromatid. But here you can see that now 1 chromosome consists of what? 2 sister chromatids. Okay, and then we get to the what? The G phase of the cycle. After G phase, then we'll go to the what? The mitosis. Okay, all right, good. We'll go to the mitosis. Okay, good. Then followed by the cytokinesis, that the division of the cytoplasm. In most books that you read, usually they lump these two together. They lump the mitosis and then the cytokinesis together. Okay, they lump it together. Most books that you come across, they do that, and it's actually accepted. All right, great. So let's see. Let me give you this particular question. So this is the cell cycle. So the cell cycle, we have the word. We have the mitosis here with our small cytokinesis there. And then G1, S phase, G2. Or from the G1 all the way to G2, they are all interphase. Okay. And then the nuclear division, the division of the nucleus here is the word, is the mitosis. Okay. And then the division of the word of the cytoplasm, that is the cytokinesis. Okay. All right, good. And as I said, most books that you come across, they don't really indicate the cytokinesis. They put it as part of the mitosis. Okay, all right, good. So let me ask you this.
So, Doc,
yes, please go ahead.
Yes,
yes,
the GO is not part of the cell cycle per se, but it's just a portion of it where when there is no replication, then it will go back to the GO.
Yeah. So the GO is not. It's GO is like
not.
It's a permanently arrested form of G1. Okay. It's like G1 that has been so permanent. It's like a permanently arrested form. You are not going to continue your life. Let's take it if somebody's in prison. Okay, the person is still a human being. The person still has a.
Hello, my end is breaking. Is it only my end or is it a general problem?
Please, I'm sure it's your end. Okay, so so here you can see that if somebody, let's say if somebody is imprisoned, the person is still a human being. He deserves to be treated with respect and dignity as human equal. But just that the person is in confinement. So yes, so here just like somebody who has been imprisoned, okay, he won't continue maybe his day-to-day lifestyle going all the way here, going all the way to no, he wouldn't. You understand? He wouldn't continue with his normal life like this, and it's actually arrest. So it's just like S1, it's like G1 that will not continue to S phase. That is basically.
Oh, okay.
Okay, so it's like G1, but this G1 it wouldn't continue. So here it is like it's not that it's even if it's producing enzyme, it's just producing enzymes and ATP for its own survival, its own sustenance, but not because it has intention to go and divide. Okay, you understand that? To just stay like that. Okay, good.
Okay,
all right, good. So that is the cell. So when it is not dividing, like mature muscle cell and mature nerve cell, they are not dividing. Okay, they don't divide. Okay, and when they don't divide like that, they are actually forever in what? In the phase of the cell cycle, we call it GO. That's permanently arrested at what? GO. Okay, good. Please, Asanto, I can see your hand is up. Please, do you have something for? Yes. Hello. Asanto, I can see your hand is up. Please, can I hear from you?
Yes, sir. Please, I want to ask a question.
Please go ahead.
Concerning the G2, the G2, it says that the cell does check up after cell division. So I want to ask a question that if it does a checkup and do repairance and all those, and do repair of everything that is supposed to be done, why is it that there's still abnormalities when a child is given birth to?
Beautiful question. I actually expected somebody to ask this question. I expected it. Okay, that's a very nice question. Well done. Okay, Santua, yes. you will do all the check-ins and everything, but still error can occur. It's not all the errors that it can repair it. Okay, they will do, for instance, the DNA, during the DNA replication, there are a lot of mechanisms, in fact about 6 mechanisms in place, just to make sure that there is no error in the DNA replication, but still mutation occurs. There's mutation. Have you seen it? Still there's mutation. All right. Okay. So at the end of our DNA replication, the error in our DNA is like 1 in, I think 1 in a billion, 1 nucleotide in 1 billion. It's still, they will say it's not a big deal. It's still a big deal considering the size of our DNA. Our DNA is quite huge. Okay, so just one error is still a big deal. So yes, it's supposed to do all the check-ins, but sometimes some of the damages it can't repair. Okay, and the cell goes ahead to divide. So yeah, that's a very beautiful question. When we come to the DNA replication in depth of it, I think I will throw more light on that. But that's a very brilliant question. Well done. Okay, great. Still to do it, but still the error will okay. All right, great. Okay, great. Well done. Okay, great. So let me ask you this particular question that I do ask my students quite often. Let's take it this way then. So a cell cycle takes 48 hours. Okay, takes 48 hours. If it's a cell cycle, okay, it takes 48 hours. If the G1 and G2, okay, takes half of the period, okay, and the S phase takes 12, let's say 10 hours. Okay, all right. Okay, 12 hours. How long will mitosis take? Okay, let's assume that mitosis is part of the cytokinesis. Okay, please let's assume in my question, let's assume that mitosis is part of the cytokinesis. So how long will the mitosis take?
12 hours.
12 hours?
Yes, please.
Okay, let's see. Are you sure? Yes. 14
14
hours. Okay, great. So here G1 and G2 together will make what? So we have what? Remember we have G1. So the cell cycle we have G1 plus G, so plus what? S plus G2 plus M. Okay. All right. So I'm putting the cytokinase as part of the mitosis. Okay. It's going to give us our total cell cycle. Okay. So the cell cycle here. And so they told us that this and this will give us 24 hours. So 24 plus what? So that will give us a half. So that's 24 plus and then the S phase take 10 hours out. So then plus M. Okay, will give us what? Will give us 48. So when we subtract, so M, okay, so M is equal to what? 48 minus 34, and that will give us what? That will give us 14 hours. Okay. All right, good. So let me give you another question on this. Okay, let me give you another question on this. Okay, let me give you another question on this. Okay, so a cell cycle takes, okay, for 72 hours. Okay, if G1 takes 40 Okay, let's say it takes let's say 30 hours. Okay, S phase takes X phase. Okay, so S takes X hours, takes let's say X hours, takes X hours, and then G1 takes
That I think you too.
20. Oh, sorry, G2 takes 20 hours. 20. Okay, let's say 25 hours. Okay, and the mitosis, let's say mitosis takes let's say mitosis takes 5 hours. Okay, then hours, find X.
30
Okay.
12.
12.
12 hours.
X
12.
is 12 hours.
12 hours.
Good, good.
12 hours.
Hours.
X is equal to 12 hours. Great. Well done. Okay, great. So this is all that you need to know about what? About the cell cycle. Now, do you know what? So have you seen the lines that we've drawn here? These lines, we call them, they are more or less checker. They are checkpoints, okay, just to make sure everything is fine before they cross. Okay, so here, this line is a checkpoint. Okay, before this one goes, every this lines here, they are more or less checkpoints. Okay, all right, great. So that's a cell cycle. Well done, well done. Okay, good. Okay, so these are basic questions. Okay, good. So now that we know about the cell cycle, so we know that the G1, the S, and then the G2 together, they make up the what? They make up the interface of the cell. Okay, they make up the interface of the cell. So after the interface, then we move on to the what? The mitosis. Okay, great. All right. So we're going to talk about so what is so we're going to talk about the mitosis. Okay, good. So we're going to talk about the mitosis. Okay, so this is still the recap of what I did earlier on. Okay, so let me take this. Okay, good. Okay, good. So all right, so I think I can duplicate my slide here. All right, good. So today, what we're going to do is that we're going to talk about mitosis, what happens here. And mitosis is what? Nuclear division. Okay, so we're going to talk about mitosis. Okay, great. So we have the word, the nuclear division. Okay, so nuclear division. So when we talk of nuclear division, in fact, we talk about we have two main types. Okay, but we know that meiosis only occurs in gonads. Okay, all right, so we have the nuclear division. And when it comes to nuclear division, that division of the nucleus, we have the word, we have the mitosis, okay, which is the most common one, and we have the word, the meiosis. Okay, meiosis. So we're going to talk about the word mitosis. Okay, good. I think I've given you the head start on it already. All right. But before we do that, let's see the mitosis. So let me see. I want you to appreciate this picture you have here first before I will talk about it. Okay. Before I'll talk about it. Okay, great. Okay, so before that, let me give you one tricky question here. That will help me. Let me give you one tricky question here. Okay, let me give you one tricky question here. Okay, so let's say this is question 2 here. Okay, so let me give you question 3. So question 3. Okay, question 3 here. So a cell at what? At G1 contains 24 chromosomes. Okay, all right, here. Now A. How many chromatids chromatids are in the cell at G1. Okay, 2. How many chromosomes, okay, are in the cell at G, at the end of what? At the end of, okay, I can say at G2, because you know that at G2. At G2. And last one, how many chromosomes, how many chromatids, okay, are in the cell. Okay, so total, let's say, okay, the total, let me make it. Okay, so how many chromatids are in the cell? Okay, in the cell at G2. Okay, let's start. Good. Let's see. Okay, let's answer this one, and then that will usher me into the word, into the cell division proper. Okay, into the cell division proper. Okay, that will usher me into the cell division proper. Okay, take your time and do this one, please. Don't rush it. I want you to get this one, because once you get it, then I know that you are on track. Okay, good. The end of same question that you write, expect this type of questions, the application questions of this nature. Expect them. Okay, I'll be giving you more of this type of questions, application questions. Please, who has finished? Who has finished? Yes, who has finished?
Hello, sir.
Hello.
Okay, sir.
Good.
So the first one is 24 chromatids.
Okay.
The second one is 24 chromosomes, and the last one is 48 chromatids.
Okay. Yes. Any different answer from what our sister has said?
Sir, I had the same. First one, 24 chromatids. Second,
Yes.
one, 24 chromatids.
The first one you had was 12. Let's listen to other people's answer. Those who have different answer from what our sister said, yes, bring the answer out. Yeah, the first one you had was 12. Please, are you there? Hello.
Hello, I had the same answer.
24.
You are 24.
Yes, please.
Okay, so yes, anybody with different answer. I want somebody with different answer. I want somebody with different answer. Do you all agree?
Yes, please.
Okay, so
the D should be 23 chromosomes.
Okay, please explain why the first answer, why the first one is what is
Doctor, with the first one, there is no cell division at G1, so it still remains the 24.
Okay, excellent.
It doesn't replicate, yeah, so it's still 24.
Hey, someone's mic is on, oh, please. Someone's mic is on.
Okay, go ahead. Excellent. Please explain the second one too. Hello.
To me, the second one.
Explain the second one.
At the G phase.
So the second one.
The G phase is set as a checker. and normal chromosome should be 23. So to me,
Hello, I cannot hear. Please,
the
is
G
it like
phase
that for everyone?
makes the chromosome to become 23.
No, I didn't say the normal chromosome I gave you is 24. I gave different organism. I didn't say normal chromosome. I just said 24 chromosomes. I didn't tell you the organism, so you wouldn't know. So it is 24. It is 24 here. I gave you 24 chromosomes. That the organism is having 24 chromosomes. Okay, and I'm asking,
Okay, sir.
how many? Yep.
Then it's 24.
So here is 24.
24.
No.
Hello.
Hello.
Okay, sir. Hello, sir.
Hello, sir.
Go ahead.
B will be 48.
Yeah, how many chromosomes? B is also 24.
Sir, B is
Is
24.
there no
And then here, 48.
Is there no replication at G2?
Yeah, so there's duplication. But when
there's no S.
It's okay.
B
So
is
please
48.
allow me explain. Please allow me to explain to her. So the G1, the chromosome has not replicated yet. So it's 24. And remember, in G1, a single chromosome consists of what? A single chromatid. A chromosome consists of a single chromatid. But at the end of S phase, when you get to G2, because that cell had what? Because the DNA has replicated before it gets to what? It replicates before it gets to G2. At the G2, you can see each chromosome is what? It's consisting of what? Two sister chromatids.
Two sister chromatids. Yeah.
So at the end of this phase, now the chromosome is not looking like that, but they are looking like this. So you can see that each chromosome now, okay, there are still one chromosome, but this one chromosome is now consists of what? Two sister chromatids. Okay, so here you can see it was one. So this is more or less G phase. So if I should draw something here like that, okay, if I should draw something here like this, so maybe this particular line becomes the what? The S phase. Okay, so after the S phase, this one okay, so let's say this is my S phase, all right, like that. So after S phase, this one is going to double up, but when they double, it doesn't separate. It's still one chromosome, but one chromosome consisting of what? Two sister chromatids. Okay, one chromosome consisting of two sister chromatids. So with that question, where's my question? Okay, so with that, how many chromosomes are there? So the chromosomes are still 24. Okay, but how many chromatids are there? The chromatids are now what? 48, because now each chromosome has two sister chromatids. Okay, beautiful. Okay, great. So that is that. Now we are going to talk about what? So the next thing that we are going to talk about, okay, so now that you know of this one, now we are going to talk about what? We are going to talk about
Hello, Doc.
Yes, please.
Please, is the meeting being recorded?
Yes, it is.
Oh, okay. Thank you.
All right. So we have the word. We have the mitosis. Okay. So mitosis is, remember, mitosis is basically it's a nuclear division. It's the division of nucleus. Okay. Good. And so before the nucleus will divide, we have the interface will occur first. And the interface is the way the DNA is going to make a copy, metabolism, ATP production, protein and enzyme is being made, and stuff like that. So after all these ones being completed, from G1 all the way to G2 being completed, then we are going to have the mitosis going to take place. Okay. Now, so what is this mitosis all about? What is this mitosis all about? Okay. So let's see, can I guess? Okay, great. What is this mitosis all about? So if you look at my picture here, this is the more or less the summary of mitosis. Okay, so you can see mitosis involves what? So let's see the general thing about mitosis. So it's what? It occurs occurs in body cells. Body cells. And body cells is also known as what? The technical term is called somatic cells. Somatic cells. Okay. So almost everybody, every cell in our body, okay, almost every cell in our body tend to undergo what? Replication. So all the body cells, okay. So it occurs in what? In body cells. Okay. And what else again? It produces what? It produces two daughter nuclei. Okay, two daughter nuclei. And those daughter nuclei, what is special about them? They are genetically identical to each other. Okay, let's say to the parent nucleus. Okay, all right, and identical to each other. All right, so they produce two daughter cells, and these two daughter cells, they are genetically identical to the parent. So the way this one here, this parent here is the parent nucleus that is going to split to become this. You know, funny thing, I've heard of, in fact, I was in one school some time back, okay, and I was standing in the window and a teacher teaching this one and says that after the division, the parent one will also be maintained and you get 3 cells. And I was in the window, I was like, hey, what sort of DDT is that? Okay, all right, it is this one. It is the same parent one. This is the parent one here. That will undergo these phases, okay? That will undergo these phases, okay, and divide into 2 daughter nuclei. All right. So it will produce what? It will produce it produces 2 daughter nuclei that are genetically identical to the parent and also identical to each other. All right. Then we have what? We have apart from this one, so you remember only 2. So here, okay. So the emphasis here is what? Genetically identical. They are genetically what? Identical. All right, good. And then they occur where? They occur in the body cells here. Okay, or somatic cells. And what other points here we talk about? Okay, another point we can talk about here is that okay, if you look at what is here, okay, apart from the fact that they are genetically identical, they are producing that, look at the chromosome number. Look at the chromosome number. If you look at the picture that we have here, the previous picture that we have here, so this is the parent. This is the parent. This is the parent. Look at the parent here. Okay, move this way here. This is the parent here. Okay, after the growth of this one, now it is here like that. And this parent here, at the end of the cell division, look at what we are getting here, the 2 daughters. So this is the parent. You see everything double up, the DNA double up here. Okay, and then, okay, so it double up at the end of what? S phase, and then move to this side like that. Okay, now look at that. Look at what is here, the chromosome here, and look at the chromosome in the daughter nuclei. What can we say there? What can we say there? You can see that the word, the daughter nuclei, okay, contain the same number of chromosomes, the same number of chromosomes as the parent nucleus. Okay, so what it means here is that if the chromosome number of the cell, if the chromosome number is 24, at the end of DNA replication, you will get what? You get each daughter cell will get 24. And do you know the reason? You see, the reason is that before the cell will divide, the DNA will replicate. The chromosome will double up, okay, like that. You see, so you can see that at the end of the day, they will come back. Okay, so look at that. Look at this one. So here, you see, before the start of the word of the nuclear division, the DNA will double up. Okay, and when it doubles up, look at how it is now. Okay, the DNA will double up. Now you can see that this one here. Okay, you can see each chromosome consists of what? 2 sister chromatids. Each chromosome consists of what? So you can see each chromosome looks like this now. Okay, so the starting point, the chromosome looks like this. Okay, this is how it looks like. Now, after going through S phase, after S phase, okay, now the chromosome becomes this. Okay, all right, to make a copy of itself like that. So each chromosome now consists of what? 2 sister chromatids. Okay, good. So here the daughter, the chromosome number you are going to get, okay, at the end of the division, the nuclear division, the daughter nuclei will contain the same chromosome number as the parent nucleus. Okay, so at the end here, like that, you can see that at the end here. So if my chromosome, if the nucleus, if the parent nucleus contains 46 chromosomes, okay, chromosomes, all right, after mitosis, each word after mitosis, each daughter cell will have what? Will have 46 chromosomes as well. We have what? 46, okay, chromosomes. Okay, good. That is so. And the reason it's like that is because the chromosome will double up. And so when they separate here, separate and that. Okay, beautiful. So now this is the overview. So at the end here, so from here up to this point here, up to this point, up to here, this is the word, this is the division. What is here is the division of the word, of the nucleus. So it is after the division of the nucleus that the cell is going to divide. Okay, and the division of the cell is called what? The division of the division of the cytoplasm is called cytokinesis. Okay, all right, so this is the overview of what? So here, the most important thing is that you know that the chromosome, okay, the chromosome number is what? Is maintained. The chromosome number is maintained. Okay, the chromosome number is maintained. All right, great, good. Now let's go to mitosis. Okay, involves 4 stages. Okay, and what are the stages? The stages are here. So the stages are the what? The prophase all the way to where? Prophase all the way to the telophase, like that. Okay, all the way to the telophase, like that. Okay, good. So the prophase all the way to the word, the telophase, that is the word. So here, more, this one is like the cell dividing. So this is like cytokinesis here. All right, good. So we want to take the stages one by one, and then we'll talk about them. Okay, we're going to take the stages one by one and talk about them. So this is the overview of mitosis with a summary of it here. Okay, good. So this is overview of mitosis with a summary of it here. So we're going to take the stages one by one, and then we'll talk about them. Okay, great. Now, so let's start with what? Okay, maybe before we talk about the stages one by one, let me give you the summary of what happens in each stage here. Okay, okay, maybe I can do that one here. All right, so let's see. let me bring some sort of some this thing here. Let me copy. No, let me copy this one. Okay, and bring it here. Okay, like that. Okay, like that. Okay, good. Something like that. Okay, so we have this one here like this. Okay, great. All right, so let's see something. Okay, good. So now let's do the summary of what each part. So let's start with the prophase. Prophase, okay, prophase. So prophase, okay, so here, please, the interface. Do you know where? Is there where I can chop off the interface? Okay, so let me do something here like this. Okay, the interface here is this one is not part of it. Okay, so this one is not part of it. Okay. Okay, great. So let me cancel this one out. This one is not part of it. Good. So let's see. So prophase. Okay, so prophase, a lot of activities things okay in the prophase. So let's see prophase here. So prophase, in the prophase, look at that. That's going to. What will happen is that the DNA. Okay, let's see some of the things that is going to occur in the prophase. So the DNA, remember the DNA. This condensed from early part of the condensation of the DNA is called what? It's called chromatin. Okay, when it condenses further, that it becomes the chromatid. Okay, like the chromosome form. Okay, So what will happen is that the chromatin, okay, begin to work. Okay, I can just say chromosome. Let me just go. Let me use the chromosome straightforward. Okay, so the chromosome, okay, begins to work. Begins to condense, okay, begin to condense. Okay, so so here you can see that. Okay, oh, maybe now I'm wrong. Okay, let's see. You can see that in the interface, the chromosome is not all that clear. That much. You can see the chromosome is not that clear. It's like yeah, it is in there, but it's a little bit messy. Okay, it's in the form of the chromatin. Okay, it's a little bit messy. So in the prophase, it begins to what? Condense. And okay, it will become what? Visible. Okay, condense. So if it's condensed, it means that it's becoming thicker. All right, good. And then you can see that here, if you look at my slide carefully, you can see the nucleolus somewhere. You can see, if you look at this structure here, you can see some nucleolus there, right? You can see some nucleolus there. Okay, good. Now this nucleolus will what? What the nucleolus is going to do? The nucleolus is going to disintegrate. Okay, the nucleolus. Okay, the nucleolus will what? Will disappear. All right, the nucleolus will disappear. The chromosomes begin to condense. Nucleolus will disappear. Please mute your mic. Okay, the chromosomes will what? Will disappear. And then when it disappears, what's the next thing? Then we have the what? We have the, remember, do you remember centrioles? Do you remember centrioles?
Yes, please.
Good. What is the function of centrioles? If you could remember, what's the function of centrioles? Yes.
It produces spindles for cell division.
Good. They tend to organize. They organize they organize the microtubules, okay, to start the growth of the spindle fibers. So you see that the centrioles, okay, so they move to the what? The opposite poles, okay. Centrioles move, okay, centrioles, okay, move to the opposite, okay, opposite what? Opposite poles, okay, and start the what? Start the growth of what? The growth of spindle fibers. The growth of spindle fibers. Okay, so you can see from my screen here that these are the what? These are the what I have here is the what? Is the centrioles. So they will move to the opposite poles. and start the growth of what? Spindle fibers. And remember the spindle fibers, we use a what? We use a microtubules, okay, microtubules, all right, to form the what? To form the spindle fibers, okay? Don't forget that. All right, good. And then, okay, so then what will happen? Have you seen the nuclear envelope here? Have you seen the nuclear envelope here? Look at that. You see, so you see early stages, you see, have you seen that? So they will start the growth of the spindle fibers. Okay, start the growth of the spindle fiber. You see the nuclear envelope in the initial phase, you can see the nuclear envelope, but right now you can't see the nuclear envelopes anymore. So the nuclear envelope will do what? So the nuclear envelope, okay? The nuclear envelope will also what? Will also disappear, will also disintegrate, okay? So, all right, so here, so 4 main things that was going to happen. Our chromosome will condense, the nucleolus will disappear, the centroles move to the poles and start the growth of the what? Spindle fibers, and then the nuclear envelope will disintegrate. Please, okay? When they ask you to describe mitosis, well, you can be asked to describe mitosis, just the process of mitosis, okay? With the aid of diagram, describe the process of mitosis. That's a very beautiful question. Please, these are the 4 key points you need to talk about. When you read stuff from book, you can get a lot of detail. There's no need for you to go into early and then the late. Just give me one, okay? Like that. So the prophets here, if you look at the prophets here, okay? If you look at the prophets here, all right, these are prophets. Okay, so the prophets here, the chromosomes will condense, okay? So the nucleolus the nucleolus will disappear. The centroles move to the poles and start the growth of spindle fibers. Okay, and remember, this spindle fibers is the word is forming from the word the microtubules, and then there is what we have the nuclear envelope. Okay, the nuclear envelope, and then the nuclear envelope is also going to what? Disintegrate. So these are 4 main things that no matter what happens, you don't have to forget. Okay, good. So this is the prophets. All right. Any questions so far? Any questions so far? These are 4 key things you need to talk about and the prophets. Okay, of mitosis. Okay. Yes, please.
I don't know if my question. Yeah. Looking at the pictures, the images that is being casted on the screen. I don't know if my question is going to be fast ahead, but the last one, does it mean that afterwards, what is going to be formed will be twins, or is still an individual, but it will still have this form? The last one, that is the cytology.
There's nothing like fertilization. Please, you pay when you ask the question, pay attention. Listen to understand, please. Don't listen to respond. Okay, the 2 are different. Okay, listen. Like when we're kids. Okay, when we're kids, now we've grown. How did we grow? This cell will divide, become these 2, and then this one will become big. Then will become big like this. Each of them will become big like this. Then it will also divide, become big again like this. It will grow to become this, and will also divide again. So this is has nothing to do with twins. Okay, when you start talking about twins, you're talking about fertilization. Okay, all right, great.
Okay.
Right. So we have the word. We have the next one is the word is the metaphase. Okay, metaphase. So metaphase, please, the iconic thing you need to tell us is that you see at the metaphase, the key thing is that the chromosome, you see, before metaphase is about that time, the spindle fibers have what? Completed their work. their growth. Okay, so let me make it this way. Complete formation of the spindle fibers. Okay, complete formation of the spindle fibers. So by the time the metaphase is starting, the spindle fibers would have completed its development. So you can see that from here the spindle fibers have grown all the way here. And then what will happen? What will happen is that the chromosomes, all the chromosomes, okay, will line up, okay, will line up at the equator of the nucleus. At the equator of the nucleus. And when they are lined up at the equator of the nucleus, what are they? You see, you can see that with the word. You see, you can see that with the word. So, okay, let me show you something. Okay, I think the last time I described it, but let me take this opportunity to talk about something here. I know I did it the last time like this. Okay, if this is the chromosome like that, okay, say, come in, let me. Okay, so let's say, let me make this one larger a little bit. It's chromosome, okay? All right, so if this is the chromosome like that, okay, like this. This side here, this side of the chromosome, so the chromosome has something like this. So this is how the chromosome looks like. Now this one here, okay, and then let's say this, so this side here, okay,
Hello?
this one, hello?
Yeah, dog. Yeah, please. The complete formation of the spindle fibers, I believe it occurs before the metaphase, is that not it?
Yeah, so I said by the time,
or it occurs at the metaphase.
it's just so that's what you call late prophase. So what I'm saying here is that before the spindle the chromosome will attach to the equator, by that time there is complete. Okay, it's not. In fact, when
Okay,
it comes
so
to
it occurs at the metaphase stage. The complete this thing is completed at the metaphase stage.
yeah, that's where we can say that it is fully completed. Okay, it's not the most of the time it's not clear cut. Like it's not clear cut that the process will end perfectly and then the metaphase will start. Okay, you understand? It's not clear cut. It's enter into each other. You get what I mean? Okay, but before we can talk of metaphase, by that time the spindle fibers, no matter how they are growing, they have completed their formation. Okay, the spindle fibers would have completed their formation by the time we are talking about the metaphase starts. There's complete formation of the spindle fibers. Okay, and then let me draw this thing for you here. So here, okay, this side here, this is the chromosome. Now, if you look at the chromosome, this chromosome has a tip here like this, a tip here, and there's another tip here like this. Okay, there's another tip here like this. Yeah. All right. Okay, great. Now, the tip here, we call this one telomere. Telomere. This part, they protect the ends of the chromosomes. So we call this one telomere. And this one is also telomere. Telomere. And then here, we call this one centromere. All right, good. Now, so this is my chromosome. This is how my chromosome looks like. Now, when the chromosome duplicates and becomes something like this, okay, the sister chromatids will be like that. I didn't draw it perfectly, but I think it's okay. So let's say this is the centromere here, centromere here. Okay, there is a special protein connecting them. Okay, like that. So there is the protein connecting them, we call it cohesin. Okay, the protein connecting, okay, joining the word, joining the sister chromatids. It's called word cohesin. All right, cohesin. So there is this cohesin here. Great. Now, what's happening? So this is the centromere. Now there will be some sort of some special protein here. Okay, so let me draw something here like that. So there is this another special protein here like this, at the back here like this. So at the back here like this. Okay, where the spindle fibers are going to lock up. Okay, so the spindle fibers will lock into this ones. They will grow and lock here like that. Okay, so they will attach here. So here, this structure that I've just pointed, let me take a different color like that. So this structure that I've just picked here, this one is called kinetochore. All right, good. Okay, so this is another kinetochore there. Great. So you see that when the chromosomes, when they come here, and then they line up at the equator, you see the spindle fibers. So they get these at the metaphase, the iconic feature of the metaphase is that all the chromosomes line up at the equator, at their centromere. Okay, with the spindle fibers, so you can see the spindle fibers will come and work. So they get attached. The spindle fibers will attach to the kinetochore. You see here. Okay, so let me bring this one here like this. Okay, like that. Okay, so let's see something here like that. So the spindle fibers here, if let's say, let me give you something like blue color here like that. So the spindle fibers will grow. Okay, lock up the kinetochore like this. Okay, lock up the kinetochore. Okay, so they will grow and lock into the kinetochore here. So if this is the chromosome, that is like let me use the black one like that. So in the interface, okay, so let's say in the interface, let's say in the interface, let's say this is the chromosome. Like this. There is this special protein here. We call it what? Kinetochore. The one that is joining this sister chromatid is called what? Cohesin. This is cohesin. Cohesin. And the one here, that's so there will be this small structural protein here, we call it kinetochore. So when the chromosomes get attached to the spindle fibers, the spindle fiber will lock up this. So let's say the spindle fibers here. So let me pick a blue like that. So the spindle fibers will come, lock up here like this, lock the kinetochore like that. Okay, like this. The other one will go from here, lock the kinetochore like that. Have you seen it? Good. That's what happens. So they line up at the equator. Okay, so they line up at the equator, like you can see here, at their centromere. Okay, at their centromere. So you can see that the centromere, this is a centromere. Okay, this is a centromere. So they line, and then there's this special protein, we call it kinetochore. All right, good. So what happens at the, okay, so now let me duplicate this one. Okay, let me duplicate that. Oh, no. Let me duplicate the whole of this. Okay, good. So at the metaphase, what do we talk about the metaphase? Okay, so at the metaphase, let me bring, let me duplicate this and bring it here because I'll be using it. Okay, so let's see here. So at the metaphase, what is going to, Okay, so the second part, I'm done with these two. Okay, so the next one is the what is the anaphase. Okay, so the metaphase, the iconic thing you need to see there is that the chromosomes are what? They line up at the equator. All of them attached to the what? To the spindle fibers at their kinetochore. Okay, or at the centromere. They attach at the what? At the centromere. And this kinetochore locks up this one. Okay, so you know that this spindle fibers locks up the kinetochore, locks up kinetochore like that. Okay, attached here like this. So what is going to happen in the anaphase? You can see here. So anaphase is what? The separation of the what? Of the sister chromatid. Okay, the separation of the sister chromatid. Remember, this chromosome here each chromosome consists of 2 sister chromatids. So at the anaphase, there is going to be what? So the iconic feature of this anaphase is the what? Is the separation of the what? Of the sister chromatids. And then they will begin to work. So begin to the sister chromatids, chromatids, okay, begin to work. Chromatids begin to move to the poles. All right. Sister chromatid begin to move to the poles. Okay, great. So with the sister chromatid moving to the poles, okay, so they separate and then begin to move to the poles. So look at how did they separate. If I should bring it here, see how would they separate? So separation of the sister chromatid is basically what is going to happen. What will cause the separation? Have you seen this word? What will break? Okay. Of course, in SS class, you wouldn't mention this one. But what will break is that the protein that is holding the sister chromatids together, which we call cohesin, will break. Okay, as the word, it will break as the spindle fibers tend to pull. Okay, these spindle fibers, they lock up the kinetochore, here, and then they will pull. Okay, and then when they pull, eventually they will break the word, the cohesin here. This cohesin will be broken, and then they break the cohesin, and then they will move towards the word. They will start moving towards the opposite poles. Okay, so they will start moving towards the word, the opposite poles. So basically, this one, in the anaphase, they will break the cohesin. The sister chromatid will separate, okay, because the cohesin will be broken, and they will begin to move towards the word, the opposite poles. Right, great. And then, so the last part is the what? The last part is the telophase. Okay, the telophase. All right, telophase. What happened now? Telophase is basically the reverse. Let me put this one. Is the reverse of prophase. Prophase. Okay, so here, all right, so let's see what happens here in the telophase. You see the word, the telophase, you can see that by the time here it will arrive the word, the sister chromatid, the chromosomes into bracket, sister chromatids, okay, well, chromatids, okay. Will arrive at the where? Will arrive at the where? At the poles. Okay, so they'll all go to this point. Okay, so they will pull them. The spindle fibers will pull all of them here. This one will pull all of them. So they'll all arrive at the poles. Okay, so the chromosomes, with the chromatids, will arrive at the poles. When we arrive at the poles, what happened now? I just said that the telophase is a reverse of the prophase. So whatever that happened in the prophase, okay, in the beginning of the prophase, everything is going to be reversed. So yes, let's see what happened there. Yes. Yes, somebody should give me one. Yes.
The nucleolus will come back.
Good. So nucleolus. The nucleolus. The nucleolus. Okay. The nucleolus what? The nucleolus. The nucleolus will
appear.
Reappear. Okay. What else again? What else again?
The
what else again?
Or what?
Good. The chromosomes. Okay. The chromosomes. Chromosomes will what? Will so initially it's what? It's condensed, right? So now it's going to what? It's going to decondense. Okay. So decondense. All right. So it's going to go back to how it used to be. It's going to go back to the mess up state again, right? It's going to decondense. Okay, great. Okay, so the chromosomes will what? Will decondense. What else again?
The nuclear envelope will also reappear.
Ooh, nuclear envelope. Nuclear envelope will what? Will reappear. What else again? What else again? What else again?
The cytoplasm starts to divide.
Okay, well, that's cytoplasm division is cytokinesis. So you can see that the spindle fibers will disintegrate. Have you seen it? You see, it was during the prophase that way. The spindle fibers began to work. Okay, that's where they begin to fall. But now everything is reversing. So spindle fibers will work, will disintegrate. Okay, the spindle fibers will disintegrate. All right, the spindle fibers will disintegrate. Beautiful. That is all. Okay. Then after this one, then saying that cytokinesis was it's not truly part of it. So when you are done here, that is it. You are done.
Hello, sir.
Okay. Yes, please.
Can we say the telophase at the telophase level, it will result in the production of 2 daughter cells?
They've not separated yet.
Okay.
They've not separated yet. It's after you are talking about daughter cells. We are talking about separation of nucleus. They've not separated yet. So now they've reached. So this is the telophase. This is the telophase. So now they've reached the poles, everything. The nucleus disappear. The spindle fibers disappear. The nucleus will reappear. Nuclear envelope will reappear. The DNA will decondense and stuff like that. So it is this at the end of this, that is the telophase. So telophase, so when they ask you to describe telophase, these are the points the marker will be looking for. Okay, good. Sometimes they can ask you with the aid of diagram. Sometimes they can ask without diagram. Okay, can ask you to describe prophase. Okay, you can describe telophase with the aid of diagram. You should be able to bring this one and show it in your diagram. Okay, that is it. Good. All right. Now, so is there a question?
Is it
Please go ahead.
Is it necessary that we start the illustrations from the interface?
Interface? Not really. You don't necessarily need to. You don't need to. You don't need to. Okay. When let's say if in exams and I ask you, you don't need to. You don't need to do that. You just if I ask you to describe mitosis with the aid of diagram, you don't need to show interface. Or interface is not part of mitosis. But because we are going to describe the structure, we want you to see how the DNA, this one you see the interface. So the DNA is what? The DNA is in the chromosome is in the condensed form. So it is the prophase that it is going to condense. So there's no need to add the interface. Interface is not part of mitosis. All right. So after this one, then we are going to get what? we are going to get cytokinesis. Okay, after this one, that we are going to get cytokinesis. In fact, I need to speed up a little bit because today I can't go beyond a certain time. I have a certain meeting elsewhere that I need to rush there as well. I think I've done very well. So let me, so we're going to talk about what? Let's talk about the cytokinesis. Let me tell cytokinesis in what? Let me get image for you in plants and what? And animals, okay? So let me see if I could get a picture of it to show you. Okay, good. I've gotten one beautiful picture here. Okay, I've gotten one beautiful picture here. All right, so let's see. So after this nuclear, this one, then we have what? We have a stage, okay, called the what? The cytokinesis. Okay, that is a word. Division of the cytoplasm. Okay, the cytoplasm is going to divide. But the cytoplasm division and the cytoplasm in plants and animals, they are not the same. Okay, so I'm going to show you a picture and then I will use it to talk about okay, so let me get a picture for you. Oh no. Okay, let's see. Let me get a picture for you here. I've gotten a nice picture here. Good, so great. So let me duplicate this one and then put this picture here. Okay. Let me so here, you know, well, let me make this one blank. Blank. Okay, so we have something like this, like that. Okay, great. So we have the word. So this is afterward, at the end of the telophase, okay, at the end of the telophase, this is the telophase. The telophase has ended here. So at the end of the telophase, now the nuclear envelope is back. The words, the DNA will decondense and everything. Okay, good. Then in the case of animal cell, okay, there's formation of something called cleavage furrow. Okay, so in animal cell, okay, in animal cell, okay, there is what? There is a formation of cleavage furrow, okay, and this cleavage furrow, remember, I told you earlier on, it is the what? It's the ring of what? This cleavage furrow, it is a ring of something. I told you, who can remember? Who still remembers? It's a ring of what? Yes. Hello. It's a ring of what? Hello. Yes, ritual. Your hand is up. It's a ring of what? Yes. Let me hear from you, ritual. Let me hear from you. It's a ring of what? Yeah. Remember, I said ring of what? I said ring of. So this cleavage is what? A ring of what? A ring of actin filament. Okay, actin filament. Remember, actin filament is what? It's a microfilament. It's the microfilament, one of the cytoskeleton. So ring of what? Ring of actin filament. And what is going to happen? So at the equator of the cell, okay, so this is the cytokinesis at the equator of the cell. So there will be what? Accumulation of the what? So there will be a ring of what? Actin filament that will accumulate at the equator of the cell, and then they will pinch inward. Okay, all right. So we have they will pinch inward like that. So you can see from here. So they are pinching inward. So they will pinch inward and eventually separate the what? The cell. Okay, so they will pinch what? Inward and eventually what? Separate the cell. So in the what? In animal cell, it is what? The cytokinesis involves the what? Involves the formation of what? Of cleavage furrow. In this cleavage furrow, that's the ring of what? Actin filaments. Okay? Or a ring of actin filaments, that's what? That pinches what? Inward okay, inward from the opposite ends of the equator of the cell. All right, good. And then eventually separate it. All right, good. So you can see from this image, okay, they will pinch inward from the opposite end of the equator, and eventually they will separate it. All right, great. And then when it comes to the plant cell, it's involved the formation of cell plates. Okay, so here the Golgi body will form this special cell plate. It's going to form something like a cell wall. Okay, so the Golgi apparatus will send material that is going to form the cell wall. Okay, so send a lot of vesicles. Okay, so we'll form a lot of vesicles. All right, okay, good. So in the case of plant cell, in plant cell, okay, so for plant cell, okay, so for plant, okay, so the cytokinesis, okay, involves the formation of a cell plate, okay. And how the cell plates like? The cell plates is basically the Golgi body. So Golgi body, okay, Golgi apparatus will bring a lot of will produce, let me say, will produce, okay, macromolecules, all right, macromolecules that form the cell wall of this, okay, the cell wall, okay, all right, and then they will do the cell wall, okay, and then they will do that, and then to work these vesicles will bring the macromolecules. So eventually, we'll produce macromolecules, okay, that will form the cell wall and bud off in vesicles. Okay, that will accumulate at the equator of the cell. All right, good. So if I should go back here, you can see here. So here the Golgi body will produce a lot of vesicles containing the materials of the cell wall. Okay, so a lot of them will come here. The vesicles will come, and then eventually they will fuse. Okay, they will accumulate at the equator here, and then they will fuse to form the cell plate, which will eventually form the cell wall that will separate the two daughter cells. Okay, so this is what I could say for mitosis. Any questions so far? Any questions so far? Before I will ask you questions. Any questions so far? Any questions so far? Any questions so far? Any questions? Yes, please.
Yes, so if you are asked to describe the mitosis, will you include the cytokinesis?
Technically speaking, if I ask you to describe mitosis, you don't have to include the cytokinesis. If I say that describe the mitotic cell division, that one you need to include the cytokinesis. Have you said it? If I say that describe the mitotic cell division, that one you have to add the cytokinesis. But if I just say describe mitosis, it doesn't include it. It ends at the telophase, prophase to telophase. You are done. Okay, but if I say describe the mitotic cell division, that one you describe the nuclear division and add the cytokinesis to it, because you need that one to make a complete what? Cell division. The emphasis on cell division. So that one you need to add that. But just mitosis, it doesn't include the cytokinesis. Okay. All right, good. Now that you're okay with this one, let's see the importance. Okay. Now that you're okay with this one, let's see the importance of the word. Okay. Yes, please.
Listen. So
please go ahead. Ask the question.
Cytokinesis. The is the actin filament breaking the telophase into two daughter cells, right?
Telophase.
Yes.
The actin filament.
I want to know the peculiar thing about the telophase.
You are taking us back. Do you pay attention? Please, you know I talk for quite long. So please, sometimes consider me. I beg. Sometimes people 5 hours non-stop, literally. So please try and pay attention very well. Because we are done with telophase here.
No easy at all.
This is telophase here. I've explained this one in quite elaborate detail, so I don't know why you are bringing it here. Okay, so this is the end of telophase. The telophase end, okay? So what you are just saying here is that at the end of telophase is this. At the end of telophase, whatever this cycle here, everything that happened in prophase is going to be reversed. So we don't just jump, okay? Everything, like the way when I started this class, I started the class in transition. The last part of the thing I did with you the last time, okay, this morning, I deliberately I started with it. So more or less, we need to show how at the end of telophase, how the cell looks like. The cell looks like at the end of telophase. When telophase is completed, this is how it looks like. Then the cytokinesis, the cell will begin to divide, basically. So this one is just to let you know, just like the way we put interface of the cell here, so that you have idea of what is the previous thing looks like before the prophase starts. So basically, this one here, the telophase is not part of the cytokinesis, but we place them here so that you have idea of how the cell looks like at the end of what? At the end of telophase. So some of the books you read, okay, some of the books you read like that, they will tell you that some of the books, they will add this point like this, that, oh, it initiates here. They will add like it's telophase what? The initiate cytokinesis. Some books will add that, okay, like that. If you don't add it strictly, I don't think you have to use any maths, okay, for that. So I want to let you know at the end of telophase what you expect. And then this is the cytokinesis. In the case of animal cell, formation of what? Actin ring. In the case of plant cell, formation of cell plate. That is all. Okay, great. Now let's move on to significance of mitosis. Okay, importance of mitosis. Okay, yes, we need to speed up a little bit because I won't go beyond 9 o'clock at all today. Okay, good. Yes, please. What are the significance of mitosis?
It leads to growth.
Okay, great. So significance of mitosis here is that one. Okay, we can talk of what? Okay. Well, before that, the take-home message of mitosis is that the daughter cells, okay, the daughter nuclei, they are genetically identical. Okay, so here they are genetically identical to that of the parent. Okay, so here they are what? So here, and then the chromosome number is maintained. Okay, chromosome number is maintained. Don't forget that chromosome number is maintained. So here we can say growth. Okay, growth and what? So growth. So when you are growing, okay, growing, for instance, embryo, okay, after the formation of the zygote, all of us, we used to be what? Embryo. Okay, so the embryonic development, okay, let's see, embryonic. Let me get you some sort of embryonic development so that embryonic what embryogenesis. Let's see. Okay, so let me show you a picture here like that. Okay. Okay, let's see. Okay, great. Let's see something like this. Okay, great. Let me bring it for you to see here. All right. Okay, let's see. Okay. So for what? For growth. Okay, so look at that. So you see, this is during the formation of the embryo. Okay, so you can see from here. Look at that. So this is the zygote. Okay, so after formation of the word of the zygote, look at that. The zygote will divide into 2 cells, will divide into 4, 4 will divide into 8, 8 to 16. Okay, 16 to 32. The 32 is the word, is the morula. Okay, so from this one, okay, all the way. So this is basically cell division. Okay, so growth. All right, good growth is there. Okay, growth is there. Okay, and then we can even add it as separate. We can even add it as separate. So embryonic, okay, embryonic development. So embryonic development is purely mitosis. After the formation of the zygote, you see, you can see the cell, each cell divided into 2 here. Okay, 2. After the zygote divided into 2, you get what? 2. So the 2 will divide into 4. 4 will divide into 8. 8, 16, 16 to what? 32. That's a morula. Okay, great. So apart from growth, we have also, they also involve in what? Embryonic development. Okay, okay, in what? In sexual reproduction. Okay, so in sexual reproduction, okay, embryonic development in sexual reproduction. Okay, great. All right, what else again? We can also look for repair. Yes, please. Go ahead with your question. Now time. Go ahead.
Please, I want to ask if we can say it results in cell multiplication.
Yes.
Cell multiplication.
So instead of saying cell multiplication, why don't you be specific?
Increase in cells.
Why don't you be specific? If you just say cell multiplication, you've not said anything, because meiosis to the cell will multiply.
Sir,
okay,
can I point it to say increase in number of cells?
So why don't you say embryonic development? Because if you just say, if I'm marking and you write increase in number of cells, I'll mark you wrong, because there's nothing technical about it. There's nothing. If you say increase in number of cells, what have you said? You've not said anything. Why don't you say growth so that you are free? Rather than say increase in number of cells. Because if you say increase in number of cells, there's nothing technical about it. If you say growth, everybody will know. But if you say increase in number of cells, meiosis to the number of cells will increase. Okay, just say growth and you are fine. Okay, all right, good. So embryonic development, and then the next one we can say of what? Repair. Okay, for repair. Okay, good. So especially when you get wound on your body or something like that. Okay, so the way when you see that when you get a cut on your skin, the skin will undergo, the skin cells will undergo mitosis and eventually repair the skin. So for repair. Okay, good. And then another thing is
sir, sorry, please, with this point, how is it different from tissue maintenance?
Repair can also be known as, well, repair means that there is damage. Okay, there is damage and then you are restoring it. That's repair. So tissue maintenance is like, I can maintain tissue without necessarily repairing it. When you say you are repairing something, it means that there was a damage and then you are restoring it. Is that not it? So let's say I've cut my skin, there's a cut on my skin. Maybe I've gotten some wound. So the cells there will undergo mitotic division, and eventually they will produce similar cells as the one that left. So that is repair. Okay, good. Well, we can add it to it. We can add it to it, but it's no tissue maintenance.
Sir.
Well, I will still keep the repair for now. Okay, that's the one I'm very sure of. Okay, all right.
Yeah, Doc.
Yeah,
Doc.
yes, please. Please
Can
go ahead. We don't have time. Please go ahead. Hurry up, please.
Yeah, can we say repair of worn out tissues?
Repair. Okay, good. Okay, good. That's being set. Worn out tissues. That's correct. Okay, repair of worn out. One. Okay, one. My brains are even tired. Worn out tissues. Yeah, that's very specific. That's good. Repair of tissues. Repair of worn out tissues. Okay, yeah. Okay, good. That's correct. Okay, repair of worn out. That's very specific. Okay, great. And then the last one that I'll add is what asexual reproduction. Asexual reproduction. Okay, so that is a asexual reproduction. So for instance, we have some organisms that they reproduce asexually. For instance, if you take something like yeast. You see, if you have yeast, you see yeast like that. Okay, you see the yeast structure like that, and you see the young yeast will bud off like that. Okay, good like that. Okay, so we'll be budding off from the parent one like that. Okay, so asexual reproduction like what? Like budding. Okay, like that is what. So maybe like budding in yeast. Okay, great. So in this case like that, so growth, okay, necessary for embryonic development in sexual reproducing organisms. And then we can also talk about what? Repair of worn out tissues. Okay, so anytime there's any damage anywhere, it will repair it. In asexual reproduction, you remember in asexual reproduction, remember the word, the daughter word, the daughter cells are genetically identical to the parent cells. Okay, they are genetically identical to the parent cells. Genetically identical to the parent cells. All right, great. Good. So this is what we can say about mitosis. The take-home message is that the number of chromosomes, okay, take-home message is that, let me go up to my summary. It occurs in somatic cells. They produce two daughter nuclei that are genetically identical to the parent nucleus and also identical to each other. The chromosome number of the parent is maintained. So if the chromosome is 12, then when after the cell division, each daughter cell will also have 12 chromosomes. And that actually accounts for genetically the genetic identification. That's what they are genetically identical. Okay, great. So let me give you this question to see whether you really understood all that I talked about, and then we can call it a day. The next time, God willing, when we meet, we'll talk about meiosis, and then so let's see. Okay, so here my questions for you is this. I wouldn't give true or false. I'm going so a cell. Okay, so a cell has a 24 chromosomes. Okay, now let me change this one because let me use human beings. So 46 chromosomes as the word at the okay, at let's say G1. All right, good. Okay, G1. Okay, do you know what? A cell has 46 chromosomes. Please stop. Okay, A. How many how many chromosomes and chromatics are in the will be present at G1. Okay, B. How many chromosomes and chromatids will be at G2. C. How, okay, if the cell undergoes mitosis, okay, if the cell undergo mitotic cell division, okay, mitotic cell division, how many chromosomes okay, chromosomes will be in each daughter cell. And the last one, how many cytokinesis will occur? All right. Okay. And then, okay, how many cytokinesis will occur? Okay, then my last question. If the daughter, okay, let's say my last question here, okay, let me put that one here. Okay, so you start work with this one, and my last question here, so this is question 1, question 2. Okay, so the question 2 is this. So question 2 is that a cell has 18 chromosomes. Okay, a cell has 18 chromosomes. If it undergoes 2 mitotic cell divisions, okay, how, if it undergoes 2 mitotic cell divisions, okay, how many daughter cells will be produced in total? And then how many chromosomes will be produced in each daughter cell? And then how many chromosomes will be produced in total, and finally, okay, finally, how many chromosomes will be produced here? Finally, how many cytokinesis, okay, kinesis will be produced? Okay, how many cytokinesis will be involved? Okay, so do it quickly and then we call it a day. And half down wall. So tired. Okay, please finish these two questions and let's call it a day.
So,
So you see the type of question I have on my screen, they are application questions. They are not just reading, surface reading stuff. You can't solve this question with surface reading. So you really have to understand the concept very well. And then okay, so good. So solve these questions quickly and then we can call it a day.
Hello, sir.
Hello.
Can I answer the first one?
Please go ahead. Please go ahead, please.
Okay. So with the question one, the A, there's going to be 46 chromosomes and 46 chromatids.
Okay, so so let me duplicate this slide here. Okay. Okay, so let's see. All right, so let's see. Number one, 46
chromosomes and 46 chromatids.
Let me duplicate the slide so that I can use it. Okay, good. Okay, so so number one, 46 chromosomes and 46 words. So 46 chromosomes, okay, chromosomes, and then what? And 46 chromatids. Okay, good. Any counter, any chroma?
she's correct.
Any counter version?
No, sir, she's on track.
Okay, go ahead. Okay.
And the B, at G2, there's going to be 46 chromosomes and 92 chromatids.
Any counter opinion? Any counter? So 46 chromosomes
and 92 chromatids.
Why? Why your answer? Can you give reason why?
So after the DNA replication at the S phase, each of the 46 chromosomes now consists of 2 sister chromatids. Therefore, there are 46 chromosomes and 92 chromatids.
Okay, good. That's a beautiful answer. That's a beautiful answer. I'm not surprised where it is coming from. I'm not surprised. I know the ladies in this class. I'm not surprised. I'm not. Okay, some are saying that, some are only adding base. Yeah, she's right. Meanwhile, the answer they had is different. All right.
And
Next one.
Next one, each daughter cell will have 46 chromosomes.
Okay, so each daughter cell will have. Okay, good. So each daughter cell, good. Each daughter cell, so we'll have 46 chromosomes. Good. Okay, good. Well done. And then the
And
last
the
one.
last one, only one cytokinesis will occur.
Good. Hmm. The ladies in this class now, they are something else. They are too good. I'm not surprised. I'm not. Okay. There, one man. You see, 4 questions. The man, you've seen how.
Asanzo, which center are you?
You see,
I'm at Pastor.
you are now asking. You are now looking for a steady partner.
I'll take your number after here, okay?
Okay.
All right. Please, I want one man to answer question 2. Now, who will man up? Silence in heaven.
I'll take question 2.
Okay, one man. Who is that? Let me see the brave man. Who is that? Who is going to take the question 2? I want to see that. Is that Aaron?
Sir, it's me. Yes, sir.
Hey, Aaron. Okay, Aaron. The priesthood. Come and lift your rod. Let's see. Go ahead.
Okay, sir. The daughter cells. The daughter cells will be 4.
The daughter cells. The daughter cells will be 4. Okay, let's see. Okay, let's see. Here. Oh, what is happening? Okay. So 4. Good. So 4. Good. Go ahead.
And the number of chromosomes in each daughter cell is 18.
The number of chromosomes in each daughter cell, 18. The man is doing it. The man is on track. The brotherhood is watching with all their eyes.
And the total number of chromosomes will be 72.
The total number of chromosomes. So that would be 18 times what? 4, right?
Yes, please.
It's 18 times 4, 72. 36 plus 36, 72. Yes. You're right. The man is gathering, kneeling it. Swan. The brotherhood is watching.
The D aspect is 2.
And the number of cytokinases is 3.
The number of cytokinases is 3. Okay. Any counterversion to the answers?
It should be 2.
No, it's 3.
No, it's 3. It's 3. It's 3.
We are even ending the class with one man down. Anyway, it's 3.
It's 3.
See, that's how we go like this. Let's see, let me draw. So this is the original cell. And so first of all, divide into 2 daughter cells. So here, and then this one will also divide. 2 mitotic division. So this one will also divide. And this one will also divide. Well, I said 2 mitotic division. So this is the first mitotic division, second mitotic division. So here, there will be one cytokinesis here, another cytokinesis here, and the first one here. So 1, 2, 3. Okay. So the cytokinesis is going to be what? So I think he has done very well. Well done, well done. Proud of you. Okay, good. So this is how far the Lord has brought us. Make sure you read about the meiosis. We will try. Okay, but tell your colleagues, the genetic class that I will have with you, I will meet the entire class, the whole of the class. The genetic class, it doesn't matter which group they are. Okay, it doesn't matter which group they are. When I'm doing the Mendelian genetics, that one I will have all the group here. Tomorrow, please try and come out. This one I finished the questions. The one that I've not finished is the data collection. So where I'll finish tomorrow, that is where your quiz will cover. So there is the data collection that I've not finished setting the questions. So data collection tomorrow, please, those of you who have been coming late, I'm begging you, tomorrow we are starting quite early, and I need to finish that so that I can finish my data collection questions and send it, okay? All right, thank you very much for having me. Have a good evening, and God bless you. Thank you. All right, so I'm gonna send
can
this.
we have this slide?
Yes, I'm gonna send it to you right now.
Thank you, sir.
Okay.
And this morning's own.
This
Please
morning,
add.
I think I did. I sent it.
No, you said you add.
No, I'll add. Yeah,
No,
I've
we said
added.
we
I
added.
continue. Yeah, I continue. So I continue what is there. So I'm going to send it to you right now. Okay, so let me see the WhatsApp. Good. Okay, so I'm gonna add it to it and then I'll send it to you right now. Okay. Good. I have to make some call. Okay, so all right, so please make sure you make time to read about the meiosis. Okay, so technically what is left is the structure of DNA replication. Okay, so let me tell you what is left. So that in your free time, you can, I'll add, let me add it here so that in your free time, then you do that. Okay, so it's left with meiosis, structure of what? DNA, and then DNA replication, and then the Mendelian genetics. It's quite a lot, okay? All right, most SS teachers don't teach this one, so we have to be really careful to get this thing sorted. Okay, so Mendelian genetics. Genetics. Okay, so in your free time, try and then read on this one. So, okay, God willing, we'll continue from here. Okay, all right, let me see the WhatsApp you're seeing. So, okay, Council of Elders, where are you? Okay, excited, good. So, let me send it this. Okay. Great, good. Okay. All right, so this is that. Okay. So please have sent the slides. Okay, so I think you'll be fine. Okay, so Council of Elders, please kindly send the slides to your colleagues.